All right, we are now moving away from the molecular side of DNA and finally getting into genetics. So let's dive in. Here are a bunch of your key terms. So genetics is just the understanding of how traits get passed on from their parent to their offspring. It's how we take our genetic code, mix it with another organism's, and create a new being with its own unique genetic identity. So when we're talking about traits, traits are just characteristics that can be inherited. So not everything is DNA based pertaining to who we are. There's a lot of aspects of personality, intelligence, and things like that that do not have a genetic basis. These are things that are environmental solely, or partially genetic, partially environmental. So traits are the ones that get inherited, the things that get passed down to the next generation. So genetics is really asking the question, how do our genes, how does our DNA get inherited? And can we potentially predict what we can see in future generations? So if we remember from our talk about chromosomes, we get half of our chromosomes from each parent. So we have two copies of every single chromosome, one whole copy, one set we get from one parent, one whole set we get from another. Now, this is for sexually reproducing organisms only. We're not talking about asexual reproduction here, purely where two organisms sexually reproduce and produce an offspring. So when we talk about the variations between organisms, remember that they are due to the variations of the order of our bases. So a gene will be almost the same for every single human, let's say the gene for eye color, but there may be a few A's, T's, C's, and G's that are slightly different from one person to the next that's going to determine whether you have brown eyes, green eyes, blue eyes, hazel eyes, etc. So that variation is all based on the order of the nitrogen bases in our DNA. So keep that in mind as we move forward. So this all starts historically with Gregor Mendel. He was a monk, lived in the 1800s, came from a large, poor farming family. He wanted to be educated, and at the time, the only way to do so was to attach yourself to the church unless you had money. So because his parents couldn't afford to send him to school, they said, go become a priest and they will teach you and educate you, and then you can work and be a teacher for them. So he became a priest and he lived and worked in a monastery. He both taught at the monastery and he cared for the garden because he came from a farming background. Now, something really interesting about Mendel's work as well. First, look at the time period. This is before we discovered the structure of DNA. This was when we were still really developing cell theory. So he made all of these observations and discoveries before we even had an understanding of DNA. The other interesting thing was when we were discovering DNA, Mendel's work had been lost to time. A lot of it had been destroyed by the monks, and otherwise it was just kind of hiding out in their library and not really being published and sent around the world. So these ideas of genetics had kind of hidden for at least a half century and were rediscovered right around the time when we were discovering the structure of DNA for the first time. So kind of a really fortuitous series of events when it comes to Mendel and our understanding of genetics. So when Mendel was working in the garden, he performed a series of experiments on the plants and on some of the animals as well. He was working with his bees, he was working with little mice and rats, and the priests ended up putting a stop to that. They said it was unnatural for him to be studying the animals, but he could study the plants. So most of his work that survived, especially, is related to his studies on plants. And he was looking at how these traits were inherited by the offspring, the children of specific plants. Now, most of the stuff that we know about Mendel comes from his work on pea plants. And peas have many different varieties and many different traits, But these traits are all very specific and they're very easily studied. Honestly, it was one more piece of fortune or luck that he chose the pea plant to do these studies on, because the pea plants are quite simplistic in how their genetics work. They have basically two different versions of their specific genes. So when we talk about the seeds, their seeds could be round or wrinkled. Their interior could be yellow or green. Their flowers could be white or purple. There was no mixing or blending of these traits. They were all very specific. You got one or the other, and it was very obvious which one you got. Things weren't hiding. And these were simply inherited traits. We're going to talk a little bit more in class about how complex genetics can become, but these are very straightforwardly inherited. So these were the traits that Mendel looked at in his piece. And when he was doing his experiments, he started with what were called true breeding plants. These are plants who had the same traits as their parents. So a purple flowered plant, both of its parents had purple flowers. A white flowered plant, both of its parents had white flowers. That's what we're talking about here. So he took the pollen from one plant and transferred it to the other plant to cross them or to breed them. And then what he would do is he would collect the seeds that were produced from this cross of plants, and then he would plant all of the seeds and see how they turned out. So this original generation that he cross-pollinated with the true breeding plants, we call that the parental generation. And he liked to look at two opposite traits, so purple flowers versus white, tall versus short. He would pick one of one trait and one of the other trait. So that is the P, parental generation. And what he did was called a genetic cross. The results of this genetic cross produced the filial one generation. Filial just means child or son in Latin. So this is the first generation of children. So we can see the two parental generation producing the F1. Now we then can cross or self-fertilize the F1 generation. That's one interesting thing you can do with plants in particular, is they can self-fertilize. So the genetics are going to be identical in both of the parents in this case, and produce the F2 generation, the filial 2. This would be the grandchildren of the P generation. So we have P, F1, and F2, parents, children, grandchildren. So this is how his genetic crosses went, and he would look at the distribution of the different traits in all of the offspring and the grandchildren. So what he found was that no matter what trait he looked at, when he crossed the P generation, all of the F1 had the same trait show up. So he would cross a tall or a short or a purple and a white, and all of the F1 generation looked the same. They all inherited the same trait. All of them would be tall, or all of them would have purple flowers. So he found that one trait was stronger than the others. It seemed to overpower the other trait in the parental generation. The tall overpowered the short, the purple overpowered the white. So he then would, like I said, cross-pollinate the F1 generation, so it would be a tall plant self-fertilizing with another tall plant. But in this case, even though both parents were tall, in the F2 generation, he would see this very interesting mix of the traits. This short plant would suddenly reappear in the grandchildren in the F2 generation. About 75% of the plants would still be tall, but 25% of them would receive this weaker trait, and they would be short. So we now call this stronger trait the dominant trait. The weaker or the hidden trait we call the recessive. So when you have a copy of the dominant gene, it will always overpower the recessive gene. And recessive genes can only show up when you have both copies that are recessive, when there's no dominant copy to be found. So when your mom and your dad give you a copy of the recessive, that's the only way that you can see that trait. Otherwise, if you get even one copy of a dominant trait, it's going to overpower it. So when we're talking about these different versions of a trait, there's a term for that, and it's called alleles. These are different copies, different versions of the same gene. So we're talking about hair color or eye color. It's just different versions of that same particular gene. So when we talk about the hair color gene, your dad might give you a recessive for blonde. Your mom might give you a dominant for brown. So those are the two different copies here, the blonde and the brown. Those are different alleles. So when you pair alleles, you can have two dominant traits, you can have two recessive traits, or you may end up with one dominant, one recessive, based on the number of alleles you have. Now, also, alleles aren't just limited to two. As we see with hair color, there's a lot more than just brown and blonde. There's also red, there's mix of these. For a lot of our traits, it's not just tall or short or purple or white. There's often instances where you can have many different types of alleles, but we'll talk about that in the future. For now, we're just going to limit ourselves to what Mendel is discovering with his peas, and in his case, there were only two alleles per trait. So the dominant was things like round yellow seeds, inflated pods that were green with purple flowers that were tall, and axial, meaning the flowers showed up in the middle of the plant versus on the tips. And then here you can see all the recessive versions of all these traits that Mendel was looking at. So now that we have a better understanding of DNA, we can actually give terms to these in a much better way than Mendel was able to. So the genotype is the genetic makeup of a trait. So geno for genetic, that's how I remember it. And this, we're looking at the allele itself. Are you given a dominant allele or a recessive allele? So if it's dominant, we represent that with a capital letter. If it's recessive, we represent it with a lowercase letter. And so we can have, as I said, two dominants, a dominant and a recessive, or two recessives. The phenotype is the physical appearance of the trait. Phenotype, physical, PHY, or PH for both of them. So in this case, we're not referring to the genetic makeup or the alleles. We're saying, what does it look like? Is it purple? Or is it white? Is it tall? Is it short? Are the seeds round? Are they wrinkled? Things like that. So we can look at both the genotype and the phenotype for an organism. And we can see that even if the phenotype is the same, the genotype might not be. Because in the case of two dominant alleles or a dominant and a recessive, you will end up with the same phenotype because the dominant is going to overpower that little recessive allele. And the only time you get that recessive is when you have both of the recessive alleles. So just because they look the same doesn't necessarily mean they have the same genotype. So when we talk about genotype, let's get into some of the terms that we use to describe genotypes. So the first term is homozygous. Homo means the same in science, so this is where both of the alleles are the same. You can be homozygous dominant, meaning both of your alleles are the same and they are both the dominant or capital letter. Or you can be homozygous recessive, meaning both are the same and it's the recessive or the lowercase allele. So we have homozygous dominant, homozygous recessive. But what about when they are mixed? You have one dominant, one recessive. There we use the term heterozygous, hetero meaning different. So there's no heterozygous dominant or recessive because we're just saying there's only two and they're both different from each other. The only reason why we add dominant and recessive to homozygous is to specify whether we're talking about two capitals or two lowercases. So that is how we label a genotype. And you'll have to know this for your quiz. I may give you the 2 capital letters, or I may say this organism is homozygous dominant, and you'll have to know what that means. Now, bringing this back to the central dogma that we learned about before, where DNA codes for RNA, which codes for the protein, we can still see that here. The genotype, the DNA, codes for the phenotype, the protein, the physical appearance. So this is still reinforcing the idea of the central dogma, where genotype codes for phenotype, DNA codes for the protein. And then to kind of bring this all to a close, we're going to discuss Mendel's laws. So biology doesn't have a lot of laws. Laws in science tend to deal with more mathematical explanations for things. And unfortunately, in biology, we have such complex systems that we can't really boil things down to a nice mathematical equation. We have more theories in biology, which are just equally as valid, except we have more experimental proof for them rather than mathematical proof for them. But Mendel has 3 laws, so this is quite unique in the world of biology, and for all 3 to come from a guy who didn't even know what DNA was pretty impressive. So the first one is quite straightforward and simple, and probably the most important that I want you to take away from this lesson, and that is Mendel's law of dominance. So the dominant allele, the dominant trait, is going to overpower the recessive to show up in the offspring. So when you have the capital letter and the lowercase letter, the capital letter is going to overpower the lowercase. So we only see the dominant trait. The only way that the recessive trait is shown is if we inherit 2 copies of the recessive allele, both of the lowercase, in order to see that recessive trait. Now, he also has 2 other laws. They're going to be a little bit harder to explain only because we haven't gone through meiosis in this class, but I'm going to give you the really, really quick version of this. So his other 2 laws are the laws of segregation and the laws of independent assortment. So if you think about you as an organism, you have 2 copies of all of your chromosomes, one from your mom, one from your dad. So when you go to make your sperm or your egg, you don't want to give 2 copies to your child. You want to separate those 2 copies and randomly give your child one of them. That way, in that sperm or in that egg, when it fuses with the opposite cell, it can create a pair of those chromosomes. So in this case, the law of segregation says that the 2 alleles, the 2 copies of your genes, one on each chromosome, separate when we're forming our gametes, when we're forming our sperm and egg. So that way only one is passed to the offspring. That's it. That's the whole law. The law of independent assortment talks about how those chromosomes, how those alleles are arranged when we are making those gametes. So the alleles are going to arrange themselves randomly, meaning when I go to make my sperm or my eggs, I'm not putting all of the chromosomes from my dad on one side and all of the chromosomes from my mom on the other and then separating them. I might have, 60% from my dad on one side and only 40% on the other side from my dad. So the chromosomes randomly assort themselves before they are separated. So that's what that one is getting into. The alleles arrange themselves randomly, then they get segregated or separated, and then that forms the gamete. So this kind of creates even more randomness in the sperm and the egg because it's not necessarily just copying my DNA exactly and a kid gets half of my DNA equally. Maybe one of my kids gets more DNA from my dad. Maybe the other gets more from my mom. It just depends. So those are the 3 laws of Mendel: the law of dominance, the law of segregation, and the law of independent assortment. So, as I mentioned, the peas were quite a rare find. They conform to what we call Mendelian inheritance, where it's just simple two alleles, one's dominant, one's recessive. And so Mendel got quite lucky. Some of his other experiments he got a little more confused with because they weren't adhering to the same idea because there was a little bit more complexity coming into play. And that's what we're going to talk about mainly in class. We're going to deal with, yes, Mendel's laws are true, but life likes to take the base and make it just a little bit more complicated and add a little bit of spice to it. So we're going to look at some more complex ways that our genes can be inherited. They can share dominance, they can control more than one trait, two traits can be linked and passed on together. There's lots of really cool ways that we can inherit our genetics that take into account Mendel but add to it. So Mendel isn't wrong, he's just only the base part of it. And so next we're going to talk about all that non-Mendelian inheritance. And that is it for today. Make sure you answer the homework questions, and I will see you guys later.